The next structure we're going to look at is the spleen. It's a blood-rich organ about the size of your fist, located in the left side of the abdominal cavity. It's just below the stomach. It's the largest lymphoid organ. It's served very well by the splenic artery and then drained by the splenic vein. They're going to enter and exit at the hilum. The functions of the spleen are essentially twofold, and we'll look at some expanded, we'll say, functions later on. But the main functions are really as a site of lymphocyte proliferation and immune surveillance and response, as well as the ability to cleanse blood of aged blood cells, mainly red blood cells and platelets. There also are macrophages that are able to remove cell debris. The spleen is sometimes known as the red blood cell graveyard because of that secondary function. Here in this image, we can see the spleen located slightly behind the stomach. The spleen is well vascularized by the splenic artery and splenic vein. So this location where they enter and exit is the hilum. Here is figure 19.7c. We can see the spleen, which is going to be slightly near the pancreas. You can also see the splenic artery, which is quite large for the size of the spleen. And that's because the spleen is well vascularized, but also able to receive old red blood cells and platelets and is responsible for breaking them down. So to expand on the functions of the spleen is also able to store breakdown products of red blood cells, things like iron for later reuse. We're also able to see that the spleen stores platelets and monocytes for release into the blood. These monocytes, when move into spleen tissue, can also become macrophages, which can help with the function of the spleen. We also know that the spleen is likely the site of fetal erythrocyte production, and that it's encased in a fibrous capsule. Parts of this capsule do go into the spleen, and these parts of the capsule that go into the spleen are called trabeculae. Now, based on the two functions we talked about at first for the spleen, which is immune system surveillance and response, as well as the breakdown of red blood cells and platelets, there's two areas that correspond to those functions. The white pulp, so where all the immune functions take place, and the red pulp, which is where the kind of red blood cell and platelet destruction occur. So the white pulp is the site where the immune functions occur. The red pulp is the sites where old blood cells and bloodborne pathogens are destroyed. And as a result, the white pulp contains mostly lymphocytes that reside on reticular fibers. And these white pulp areas tend to cluster around central arteries, and they tend to appear kind of as areas within the red pulp. The red pulp tends to cluster around the splenic cords and splenic sinusoids, which are associated with the venous structures of a spleen. And on the red pulp, or in the red pulp, I should say, we would find macrophages that are able to engulf or phagocytose red blood cells and kind of help break them down. So in figure 19.7b, we can see the spleen. We can see its capsule, its trabeculae. We can see the red pulp, which surrounds the white pulp. The white pulp is near the central arteries, whereas the red pulp is associated with the venous structures. Here's a histograph showing you not only the capsule, but how the white pulp resides as islands within the red pulp. The spleen can become injured because it has a thin capsule, so a direct blow or severe infection can cause that to rupture, and because the spleen is so well vascularized, that can lead to blood going into or spilling into the peritoneal cavity. Now, splenectomy used to be a standard treatment in order to prevent hemorrhaging and shock, but we discovered that the spleen can repair itself, and therefore the frequency of emergency splenectomies has definitely decreased dramatically. A splenectomy is just the removal of a ruptured spleen, but if the spleen does need to be removed, the liver and the bone marrow will take over most of its functions. And we found that in children that are younger than 12 years of age, if we just leave a piece of the spleen, as long as we leave a small part, that the spleen will actually regenerate. Next, let's look at the mucosa-associated lymphoid tissue, or MALT. This is just lymphoid tissue you find in mucous membranes. You find it in the mucosa of the respiratory tract, the genitourinary organs, and the digestive tract. Now, the largest collections of MALT are found at the tonsils, the pyrapatras, and the appendix. And we find that the mucosa-associated lymphoid tissue is able to protect us from pathogens trying to enter the body, but it's not, again, foolproof. Tonsils are the simplest lymphoid organs. They're not fully encapsulated, So they're a little different in structure. Their capsule is surrounded by some epithelial tissue, but structurally they're similar to lymph nodes in that they contain germinal centers with proliferating B cells. They form a ring of lymphatic tissue around the pharynx, which is your throat, and they appear as swellings of the mucosa. Their names are just based on their location. Their names are the palatine tonsils, lingual tonsils, pharyngeal tonsils, which are called the adenoids oftentimes when they're inflamed versus when they're not inflamed, and the tubal tonsils. The palatine tonsils are the posterior end of the oral cavity on the soft part of the palate, or the soft palate, often as it's called. They're the largest of the tonsils, usually the ones that are infected. There's the lingual tonsils at the base of the tongue, the pharyngeal tonsils, again, the ones called the adenoids located in the nasopharynx, so pharyngeal in the pharynx and the throat. And the tubal tonsils, which are near the auditory tubes or eustachian tubes. The tonsils function really close to that of the lymph nodes. They're, again, not fully encapsulated, which is different than lymph nodes, which are fully encapsulated, but both contain follicles with germinal centers and scattered lymphocytes. In the germinal centers, that's mainly B cells. But the tonsils, because of their structure, the fact that they're not fully encapsulated, they're able to trap and remove pathogens in the food and air because their overlying epithelium invaginates over the open encapsulation and forms tonsillar crypts. That means that bacteria or particulate matter is able to get trapped in these crypts, and when they're trapped, they're able to be monitored, surveyed, and then destroyed. Now, it's a little risky to lure bacteria into deeper tissues. Oftentimes, we're trying to keep pathogens out of deeper tissues, but the respiratory system and digestive system are definitely kind of the first line of defense for keeping things out of deeper tissues. So they kind of take this risk in allowing their immune cells become activated by having close contact with these pathogens. And that would allow us to build memory cells against these potential pathogens in the future. So here's the histology of a palatine tonsil. We can see openings. Those are the invaginations covered by epithelium of the oral and pharynx mucosa. And we can also see the germinal centers, which would contain proliferating B cells, which are lymphocytes. Peyer's patches are also considered mucosa-associated lymphoid tissue, and they're just clusters of lymphoid follicles in the distal portion of the small intestine. They're sometimes called aggregated lymphoid nodules, very similar to tonsils. Their location aids in their function. They're in an area where they can monitor pathogens, an area where absorption is taking place, and oftentimes we want to prevent absorption of pathogens to the blood or into deeper tissues. We want to prevent pathogens from essentially breaching the intestinal wall. And this is another area where we can generate memory lymphocytes. So here in figure 19.9 is a Peyer's patch. And we can see that there's plenty of nodules near that smooth muscle wall of the small intestine, but essentially they're in the submucosa, and these all are essentially germinal centers with proliferating B cells that can act against possibly dangerous antigens of bacteria or something else. The appendix is going to be an offshoot of the first part of the large intestine called the cecum. And the appendix is just kind of a little piece of tissue or sac that hangs off the cecum. It contains a large number of lymphoid follicles, so it's essentially like a Peyer's patch not located in the small intestine. It's also able to destroy bacteria, prevent them from breaching the intestinal wall, and generate memory lymphocytes. Here we see some of those lymphoid organs we've been talking about. So just a reminder, the spleen is the largest lymphoid organ. Then you have the thymus, which we've yet to talk about, so that's coming up. The tonsils up here, Peyer's patches, small intestine, appendix over here. So let's talk about the thymus. The thymus is bilobed. It's a lymphoid organ found in the inferior neck. It does extend slightly into the mediastinum and does overlie the heart partially. Now, it functions as a lymphoid organ usually more in childhood as opposed to adulthood, and it's where T cells mature. It's definitely most active when we're younger, so it's the largest in size during childhood. It stops growing and atrophies as we go into adolescence and then gradually into adulthood. And that atrophy does associate or is associated with a reduction in its activity. So even though it's atrophied in adulthood, it will still produce immunocompetent T cells, but it does so more slowly. So our thymus is going to be an organ located kind of in the inferior neck near that mediastinum overlying the heart. Is where T cells mature and it's most active in childhood as opposed to adulthood, where it still works but not as fast. The thymus is broken into lobules and it does have an outer cortex and inner medulla. It contains plenty of dividing lymphocytes inside of it, and those lymphocytes we know are T cells. It's where T cells mature. There's some scattered macrophages, and we know that the medulla contains some lymphocytes, but not as many as the cortex. Now, thymic corpuscles are also found in the medulla of the thymus, and a thymic corpuscle is where we would see the generation of Treg cells or regulatory T cells. Treg cells are a type of T cell that helps us prevent autoimmunity. There are also two other types of T cells, which are T helper and T cytotoxic. So Treg cells are kind of a subset of those two families. The thymus is different from other lymphoid organs because it has no follicles, because it lacks B cells. So it doesn't have any germinal centers. It's not going to directly fight antigens. Instead, it's really going to be focused on T lymphocyte maturation. It actually contains a blood-thymus barrier, which means that immature T lymphocytes are isolated from any antigens to prevent premature activation. So if something were to happen to somebody and we'll say their thymus was damaged and it broke the capsule and immature T cells got into circulation, there could be a major immune response because the body wouldn't recognize immature T lymphocytes. So that maturation process is important. Also, the stroma of the thymus is different from other lymphoid organs. It's made up of epithelial cells, whereas our other lymphoid organs, their stroma was made up of reticular fibers. But this epithelial cell stroma does provide the right environment for T lymphocytes to become immunocompetent, or basically activated and completely mature. So here's the thymus. It has a capsule with trabeculae, an outer cortex, and an inner medulla. In the medulla, there are thymic corpuscles where our Treg cells mature. And then just to overview our lymphoid organs and tissues that we looked at, in the other part of the video lectures, we looked at the lymph nodes, which cleanse lymph, and they're a site for lymphocyte activation and proliferation. We also looked at the spleen, the mucosa-associated lymphoid tissue, including Peyer's patches, the appendix, and your tonsils, as well as the thymus in this part of the video lecture. This chart is a great chart to be looking at. It tells you whether or not our lymphoid organs and tissues have a capsule, which for the ones listed here is a resounding yes. Whether or not they have a cortex and medulla, which is going to be about 50-50. We don't really see a cortex and medulla in the spleen. We do have the red pulp and white pulp areas. We don't really see a cortex and medulla in the tonsils or the appendix or the Peyer's patches. But they definitely have germinal centers in those mucosa-associated lymphoid tissues. So we can say yes that the MALT does have lymphoid follicles. Yes, that the lymph nodes have lymphoid follicles, specifically in the cortex. We even saw lymphoid follicles in the spleen, but in the white pulp, which makes sense because those germinal centers contain proliferating lymphocytes. But the thymus does not contain lymphoid follicles because it doesn't contain B cells. The stroma is for the most part reticular connective tissue, except for when we talk about the thymus, that was epithelial tissue. We had these special thymic corpuscles in the thymus where Treg cells mature. In the mucosa-associated lymphoid tissue, we had diffuse lymphatic tissue in addition to follicles. And we saw that for the tonsils, we had unencapsulation, which allowed for the generation of tonsillar crypts and the trapping of bacteria and pathogens. And then the spleen again had red and white pulp, and lymph nodes were going to contain both afferent and efferent lymphatic vessels, but more afferent than efferent in order to stagnate that lymph flow and allow the components of a lymph node to work. And that's it for chapter 19. Make sure to review both parts and look at the chapter in your lecture textbook.